Greetings to everyone. Welcome back to this channel called Learn Bio with Janet. In this video, we'll be studying Form 5 Biology, Chapter 6, and the syllabus is the KBSM syllabus, the old syllabus. So the title of Chapter 6 is Variation. It's the last topic in the Form 5 syllabus. And we're going to discuss subtopic 6.1, Variation in Organisms. All right. So we'll be discussing the tale of two moths. Just like tale of two cities. Here's the tale of two moths. And also we'll be discussing the advantages of variation and also the types of variation. The learning outcomes for this video are as follows. After this lesson, you should be able to state the importance of variation in organisms. Secondly, be able to state examples of variation in humans. Thirdly, be able to state the types of variation. And fourthly, be able to compare and contrast continuous variation with discontinuous variation. Firstly, what is variation and what is the importance of variation? Variation means differences in characteristics among individuals of the same species. We must compare the individuals of the same species first to compare the differences. For example, we humans all belong to the species called Homo sapiens. Now, if we compare a class of students, there are differences in gender, hair type, height, blood group, and so forth. Okay? For example, some students are male, some students are female. Some students have wavy or curly hair, and others have straight hair. Okay? Height, some students are short, some are of medium height, and some are taller. Now, the importance of variation is to increase the ability of a species to adapt to changes in the environment, and this will help the species to survive. So the environment is not going to remain the same forever, and from time to time, the environment will change. There'll be changes. Or a disease may arise, a new disease, for example, COVID-19. So it is up to the species to adapt itself and adjust to the changes in the environment. So some individuals in the species may not be able to adapt to the changes in the environment or may not be able to survive the disease and may die. But if there's variation, that means that there are other individuals with other traits, different traits from the individuals that died, that may be able to cope better and adapt to the changes. Okay, so the more the different traits that exist in the species, the better. Okay, and it will help the species to survive. Example, if all the members of the species were genetically identical, that means if all of them are clones. An environmental disaster or disease that kills one of them will probably kill all of them. For example, clones have the same resistance to disease. So if one of them dies from a disease, then most likely all of them will die, or most of them will die. Okay, and this we have studied from Chapter 5 of the Form 4 syllabus, right, on cell division and on cloning. So, but on the other hand, if individuals have variation, some have different traits that enable them to survive the environmental changes or the disease. So those individuals that show variation, that means they are not clones. They will have different resistance to disease. So some may die from the disease, but others may survive because maybe their resistance is greater. And this they can reproduce. The survivors can reproduce and pass down the beneficial genes to their offspring so that they can continue to survive. Now, C, a species with variation has many different features, such as a variety of colors, and thus they are better adapted to the different and new habitats to escape from predators through camouflage. Camouflage means the ability of the organism to blend in with the environment by having similar colors with the environment or similar patterns, so that they are not clearly seen by the predators, and so they can escape being eaten by the predators. And this will enable the species to survive. For example, we are going to study about the moth called Discontinuous in the next slide. So we'll discuss how the species can survive if it has a variety of colors. Let us now discuss the importance of variation to a species survival. So we're going to look at a case study involving a moth of the species Biston betullaria. This story is a true story, kisah benar, and it happened in England around the time of the Industrial Revolution, when industrialization took place in England. Okay, so I will call it the tale of two moths. So there are two variants of this moth, which is called the peppered moth, because it has specks on its body, like like pepper like that. So this moth comes in two colors. We can see there are two colors for this moth: the light gray colored moth, okay, and the dark, almost black colored moth, which we call the melanic moth, because it produces a lot of melanin, the pigment that makes the body dark, like almost black color. And this moth is caused by mutation. Because of the mutation to its genes, it produces a lot of melanin. So mutation means the abnormal changes that occur to the genetic material. So during or before the Industrial Revolution, the moth, likes to land on the tree trunks, and the light-colored moth was very well camouflaged, meaning that it had the ability to match, the colors of its body to the background and blend in well with the environment, so as to remain hidden from the predators, such as the birds who like to eat the moths. So it was well camouflaged and not clearly seen by the birds. Whereas the dark-colored moth was very clearly seen, as its colors were in great contrast to the background. Black and almost white color. Alright. So it was very clearly seen by the predators, very obvious, and the birds will more often eat this moth compared to the light-colored moth. Furthermore, there is lichen growing on the trees. Sometimes this word is pronounced lichen, sometimes lichen. So lichen growing on the trees was also very light-colored. The lichen is made up of algae and fungus that grows together in a mutualistic relationship. We have studied that in Form 4. Okay, so this lichen is also like light green color, very light color, and it matched the color of the moth. So this helped the moth to remain hidden, the light-colored moth to remain hidden from the predators. Okay, so before the Industrial Revolution, if you look at this graph of population size of moth against time in years, the light-colored moth is the one that had a bigger population size than the dark-colored moth, because it was well camouflaged, so it was not easily seen by predators and not often eaten, compared to the dark-colored moth, okay, which had greater contrast in color to the background. Okay, then the environment did not always remain like this. Because of industrialization and the building of factories in England, air pollution increased and a lot of soot was produced. So the soot landed on the trees and killed the lichen. On top of that, it also made the trees darker color. So now the situation was reversed. Because of the dark-colored bark, it is the dark-colored moth that became well camouflaged, and the light-colored moth was now exposed. It became more obvious to the predators. Okay, so the birds begin to eat more of the light-colored moths, and their population size decreased. Okay, going down here, this graph. Whereas the dark-colored moth, which was not eaten, began to reproduce more, and the population increased. As you can see here. Okay, so the situation was reversed. But they are both of the same species. So the species Biston betullaria managed to survive in spite of the change changes in the environment, from non-polluted environment to a polluted environment. Okay, because as a prey, this moth is a prey, that means it's eaten by the predators. It had to hide itself from the predators, and the way it hid was through camouflage. So because of its two colors, it is able to camouflage itself either in a light colored environment or dark colored environment. So these are the notes that you can use in the essay section, but I will not read through it again so as to save time. You can read through it when you can screenshot it or stop the video. But anyway, the first part is important. There are two variants of colors of the peppered moth, Piston betullaria, the light gray colored moth and the dark colored moth. And then from there we talk about before the Industrial Revolution in England and during the Industrial Revolution, okay, and how variation helped in the survival of this species, okay, when the environment changed color from light color to dark color. So here's a summary of our story about the moth. Now this topic is quite important because it's been tested in the structured question and also the objective question. So you must know the story of the moth, Biston betullaria. So our summary or conclusion is that variation in the color of the moth, Biston betullaria, helped the moth to adapt to the changing environment. And the darker colored moth became hidden from the predators through camouflage, so they avoid being eaten. When the trees in the background became darker due to air pollution caused by soot from the industries. Okay, so through camouflage, the black moth, the darker colored moth, was able to survive, and this led to the continuation of the species, and they avoided becoming extinct. Now let us study variation in humans. So human beings may show differences or variation in their appearance and physiology. Differences in appearance include height. Okay, some people are short, some are medium height, some are tall. Weight, okay, some people are very thin, some people are medium sized, and some people may be fat. So type and color of hair, like black hair, brown hair, yellow hair, red hair. Then eye color, for example, blue eyes, black eyes, brown eyes, green eyes, and so forth. Type of earlobe, whether free or attached earlobe. Okay, and these are all involving appearance. Another one may be the absence or presence of dimples on the cheek. That small little depressions on the cheek. Now, number 3, differences in physiology include the following: blood pressure and blood group. Blood pressure, some people have low blood pressure, some have the normal blood pressure, and some have higher blood pressure. For blood group, we have blood group A, B, AB, and O. Now, physiology involves the functions and mechanisms in the body, the functions and mechanisms of organs and cells. So the 5 types of variation in humans that we are discussing here, that are included in the textbook. Some of this is a type of earlobe, type of hair, whether curly or straight, tongue rolling ability, whether you can roll or cannot roll your tongue, thumb hyperextension, and types of fingerprints. So let's look at these 5 examples of variation. So here are the 5 examples of variation in humans, which we can clearly observe among ourselves. So the first one is the type of earlobe. Some people have the free earlobe, where there is a piece of the earlobe that hangs downwards freely, and it's not attached to the face here, to the neck. Whereas some people have the attached earlobe, so where there is no extra piece of the earlobe hanging downwards here. So usually most people will have the free earlobe compared to the attached earlobe. The second example of variation in humans is hair type. So some people have naturally curly hair, not the hair that's sperm, but it's naturally curly from young, from birth. And for others, they have the straight hair that has no curls. Okay, So we say that the curly hair is a dominant trait controlled by a dominant allele or dominant gene. Okay, we usually call it an allele. In this case, the gene is called an allele. And straight hair is controlled by a recessive trait controlled by a recessive allele. So dominant and recessive trait and alleles will be discussed at the end of this part here. Later I'll explain. Another example of variation in humans is the tongue rolling ability, as stated in the textbook. So some people can roll their tongues in the shape of a tube or cylinder, and others cannot roll their tongues at all. Now, in the textbook, this is stated as an example of variation in humans and as a type of discontinuous variation. We'll study discontinuous variation later in this video. However, as a side note, some studies have suggested that tongue rolling may not be a case of simple Mendelian genetics, as is often taught. And it seems that there are people who have learned to roll their tongues after practicing for a while. So is tongue rolling genetically determined? Or maybe the inheritance of tongue rolling is more complex? No? Is it caused by just a pair of alleles? Alleles are different versions of a gene. So is it caused by a pair of alleles, for example, a dominant allele and a recessive allele only? Or are there many other pairs of alleles that are involved? And whether it is influenced by the environment is another question. So, for example, in a study of identical twins, there are cases where one twin can roll the tongue, but the other twin cannot roll the tongue. Now, all these questions will not be discussed here, all these issues. So for exam purposes, we will just base our notes on the textbook, which says that tongue rolling is an example of variation in humans, and that tongue rolling shows discontinuous variation, which we are going to study later here. The fourth example of variation in humans is thumb hyperextension. So if you were to bend your fingers or just form the good sign with your hand, then you look at your thumb. So if your thumb were to be able to stand up in an erect way like this and doesn't bend back, this is called the normal thumb. Okay, so most people have this type of thumb. But there are people whose thumb can extend backwards. That's why it's called hyperextension. The thumb can extend backwards because they have something different about their joint here. So they can extend backwards. And this is called the hitchhiker's thumb. Named after hitchhikers that thumb a leaf from the cars that pass by. So some people have the hitchhiker's thumb. And this one there are less people with this hitchhiker's thumb. So is there any advantage to having the hitchhiker's thumb or the normal thumb? None whatsoever. It doesn't cause anybody to be better at hitchhiking. So it's just that it's a form of variation. The fifth example of variation in humans is the types of fingerprints. Alright? So fingerprints are caused by the raised ridges on the front part of the finger. So when they are raised up a bit, so if you were to press them on a stamp pad, for example, and then you press down on the paper, so the ink will on the hand will create the pattern, showing you the fingerprint. Okay, for example, the thumbprint, the fingerprint on the thumb. All right, so the first type of fingerprint is called the curve, right? Where the lines, the ridges, they start from one end and then they form a curve and then they exit on the other side, like mountains like that. Okay, that's the curve. The second type of fingerprint is called the loop. So in the loop, the ridges formed have this pattern. They form lines that move into the center and then they will curve out and exit through the same side. Okay, in and out through the same side. So That is the loop. Then the third type of pattern for the fingerprint is the whorl. So in the whorl, there are concentric circles. And right in the center, until in the center, there will be a center here. Okay? And the fourth type is called the composite, where there is a mixture of more than one type. For example, here you can see the loop, and you can see this other pattern here. Okay, so that is the composite. Now, of all these four types, the most common, according to a project that we did, I did with students in the school, most common types are the loop and the whorl. And the rare one is the curve. Okay, it's something interesting. Then, what about thumbprint? Is it the same for all people, including twins? No, twins, identical twins may have some similarities in their thumbprint, but they are not exactly the same in the patterns of on the thumb. Okay, so no two people have exactly the same thumbprint, and that's why it can be used as a method of identification. Because even for identical twins, other than genetic factors that affect the pattern of the fingerprint or thumbprint, the ridges start to be formed when the fetus is still in the mother's womb, and mechanical forces like movement of the amniotic fluid, etc., may affect the pattern of the ridges and may affect the fingerprint. Okay, that's just something extra interesting about the fingerprint. So even twins don't have exactly the same fingerprint pattern. Let us now discuss the two types of variation. So here's an overview of it. The two types of variation are number one, continuous variation, and number two, discontinuous variation. Now, in continuous variation, the differences in a characteristic are slight, meaning small, and not distinct, meaning not clear or tak jelas. For example, if you look at two people who are almost the same height, the difference in the height may not be so distinct because maybe it is only a difference of 1 cm, for example. And then there are many intermediate characteristics. For example, the height of one person may be 160 and another 170 cm, but then in between there are many different intermediate heights. For example, 161 cm, 162 cm, 163 cm, and so forth. Characteristics undergo gradual changes, moving from the shorter person to the medium height and then to the tallest person. So examples are the height of humans, weight of humans, hand span, length of foot, length of leaves from trees. All these are examples of continuous variation. Now, if you notice, all these characteristics can somehow be measured. So they are quantitative. All right. That is one way to find out if it is continuous variation. Usually, for continuous variation, the characteristics can be measured. Usually. Now, for discontinuous variation, the differences in a characteristic are distinct, or clear, or jelas. So it's very clear cut. You can see the difference. So there are no intermediate characteristics and no gradual changes. One example is tongue rolling. So you can either roll your tongue or you cannot. So there are no intermediate characteristics like being able to roll half a tongue, for example. Roll half part of your tongue. So no intermediate characteristics. So the number of phenotypes or observable characteristics are much fewer than for continuous variation, usually. Like for thumb hyperextension, you either have normal thumb or a hitchhiker's thumb. So there are only two distinct phenotypes or observable characteristics. And then, for example, fingerprints, we have 4 types of fingerprints, basically. For type of earlobe, it's either free or attached. And for blood group or blood type, we have 4 different blood groups. Now, to remember the characteristics of the discontinuous variation, for example, you can remember the 3D acronym. Like 3-dimensional, right now, 3D. But here the D stands for what? The first D is for discontinuous variation, where the differences, the second D is differences, are third D distinct. In discontinuous variation, differences are distinct or clear cut. So from there, you can give the definition for continuous variation. It's just the opposite. That for continuous variation, the differences are not distinct. Okay. So let's discuss the first one, continuous variation, in more detail. Here are three examples of characteristics that show continuous variation. The first one is height. And the second one is the hand span, and the third one is the leaf length. Okay, so for continuous variation, the differences in a characteristic are not so distinct. They are slight and not so distinct. Okay, so for example, the height may be almost similar. So the difference is not that distinct. And then, for example, one person may be 1.77 meters tall and another one 1.78 meters tall. So the characteristics undergo gradual changes, moving from one extreme to the other. Then differences between the individuals may be slight and gradual, with many intermediate phenotypes. For example, between the height of the shorter person, 1.65 meters, and the tallest one, let's say 1.78 meters, you can have many different intermediate heights. Okay, like 1.67, 1.69, and so forth. Now, examples of continuous variation are height, okay, as seen here, body weight, chest circumference, foot length, and so forth. And even the hand span too is an example of continuous variation. That means the distance between the thumb and the little finger here. Now you can measure it with a ruler. So different people have different hand span. Can be measured with a ruler. Same for leaf length. Maybe these are the leaves from three different trees of the same species. And they may show variation in the leaf length or the average leaf length. So continuous variation is quantitative in that it can be measured. The features can be measured, such as height, body weight, leaf length, hand span, can all be measured. Here are the notes for continuous variation. We have already discussed number 1 to 4, so I'll read through very quickly. Continuous variation is a type of variation in which differences in a characteristic are slight or small and not distinct, not clear or tak jelas. The characteristics undergo gradual changes as they move from one extreme to the other. There are many intermediate phenotypes or observable characteristics. For example, height, many intermediate heights. Examples of continuous variation, most have been discussed. Let's look at intelligence. So for intelligence, a person's intelligence can be measured by taking an intelligence test, such as the Mensa test. From that, a score is obtained. All right. So if we plot the number of individuals against the range of scores, just like for height, we will most probably get a graph that is bell-shaped, just like for height. Okay, and this shows that intelligence. This will show that intelligence is an example, or shows a continuous variation. It's an example of continuous variation. So number 4, continuous variation is quantitative because its features can be measured. Now let's go on to 5 and 6, which we haven't discussed yet. Continuous variation is influenced by both genetic and environmental factors. For example, the height of a person is affected by his genes that he inherits from his parents. All right, if his parents pass him the tall genes, give him the tall genes, then he may grow tall. But it's also determined by the quantity and type of nutrients in the food that he eats. Okay, so take the example of a pair of identical twins. They are genetically identical. They have the same genes, which they inherit from their parents. So one twin may be taller than the other because of the type of food that the first twin eats. For example, maybe the first twin eats enough nutrients in the food that he eats, and the other twin may be more picky or choosy and did not eat enough nutrients. Okay, and the growth may be stunted. So nutrients is an environmental factor. The type of nutrients that a person and quantity of nutrients that the person eats. Another example is skin color. So skin color is genetically determined, but also is affected by sunlight, the amount of sunlight that we are exposed to. The more sunlight a person is exposed to, the darker the skin color. Okay. Now, number 6, it is controlled by a large number of genes or many allele pairs. For example, the first allele pair, alleles here are denoted by the letters, okay? So maybe there's one allele pair on the chromosome number 5, another allele pair on chromosome number 8, another allele pair on chromosome number 10, and so forth, that determine a particular characteristic, determine height in a person, for example, that influence the height in a person. Okay, so this is what we call the polygenes. Many genes are involved to determine the characteristic that shows continuous variation, like height. Now, to explain further the last point just now, we said that in continuous variation, a characteristic is controlled by large number of genes or many genes which interact with each other to influence the phenotype. Okay, so let's find out what we mean by a gene. So in Mendel's experiment on monohybrid inheritance, in chapter 5, entitled Inheritance, we have already found out that height in the pea plant, not humans, height in pea plant is controlled or determined by a gene. And this gene consists of a pair of alleles, meaning alleles means different versions of the gene or different forms of the gene. So the pair of alleles that control height in pea plant are big T to represent the allele for tall stem and small t to represent the allele for short stem. Okay. However, for humans is different. Height in humans is controlled by many pairs of alleles, not just one pair, many pairs of alleles on different pairs of homologous chromosomes. So for example, there may be a pair of allele on chromosome number 1 that controls height, but there's another pair on chromosome number 5 also that also affects the height of a person. Another pair of alleles on chromosome number 8, for example. Okay, these are all just hypothetical, but it's just to explain the meaning of the word, the phrase many genes. So one pair of alleles equals to one gene, but there are many pairs of alleles involved. So we say that the characteristic is controlled by many genes. Now, what is the graph for a characteristic that shows continuous variation? So this type of question can come up for paper 3, where you can be given a set of values for height, for example, then you can maybe ask to plot it. Okay, so how do we draw the graph for a characteristic that shows continuous variation? First, let's say in the class, the teacher will draw up this table, all right, with the range of heights all written down, and then she will ask the students who have the range, the height that's in this range, the first range, 150 to 154 cm. So those students whose height fall into this range will put up their hands, she'll count the number of students and write it down. And then for the other values, she will repeat the same procedure to find out the number of students with the different range of heights. So after getting the statistics here, and one thing to remember is you must measure the heights or find out the heights of students from the same gender. That means all girls or all boys. We cannot mix them up because the values of height for the boys are different from that of the girls. All right, if you mix up the gender, you may not be able to get the graph that is typical, okay, for continuous variation. Okay, now, so after you have written down or filled up this table, it's time to draw the graph of number of individuals against the range of heights. So you may have plotted this type of graph in maths before or at maths. So first, for your manipulated variable here, it should be the range of heights. So you must write it down here. Here 150 to 154. Okay, I didn't write down, but you have to write down. Then here 155 to 159 and so forth. Okay, and then you fill up the graph by drawing the bars for the different number of individuals for each range of height. Okay, draw bars. They're of same width. Okay, and the bars must stick together, not separated. Stick together so that you get a histogram. So that you can get a histogram. Then find the middle point of each bar and join them together using a pencil. You draw and freehand. Draw by freehand. Don't use a ruler. Join the center points together to get a smooth curve. Draw a smooth curve. So you will see that it is a bell-shaped curve, which we call a normal distribution curve. Now, what are the characteristics of the normal distribution curve? Most of the individuals will be at the mean, the middle. For example, the range of heights that is 160 to 164 cm. These have the most students. Okay, most of the students will fall or be in this range of heights. So that will be the highest point. And then there are fewer individuals at the extremes, meaning the shortest individuals and the tallest individuals. There are less of them. Fewer. There are fewer of them. Okay, so that's how you get a bell-shaped curve. And this type of graph is called a histogram. That is for continuous variation. Let us now discuss discontinuous variation, the second type of variation. So in discontinuous variation, the differences in a characteristic are distinct or clear or jelas, with no intermediate characteristics. For example, in the ABO blood group system, we have 4 blood groups, A, B, AB, and O. And by the way, the presence of antigens is what determines the blood group of a person. Those in the blood group A, with blood group A, for example, have antigen A on the surface of the red blood cells. Okay, now for types of earlobes, there are 2 types. Either the free earlobe, which hangs downwards and is not attached to the face, and then you have the attached earlobe. Okay, for tongue rolling, a person can either roll his tongue or cannot roll his tongue. And for presence of dimples are lesung pipit, called lesung pipit or lesung pipi in Malay, or ciwo in Mandarin. Okay, so a person either has dimples or does not have dimples. There are no intermediate characteristics here. They either have or they don't have it. Okay, so there are no gradual changes in the characteristics, just distinct categories. Now let's discuss the notes for discontinuous variation. Discontinuous variation is a type of variation in which differences in a characteristic are distinct with no intermediate characteristics. Individuals either have or do not have a certain characteristic. There is no gradual change between the 2 extremes. So it's very straightforward. Now the characteristics fall into distinct categories, usually very few categories, like 2 or 4 categories like that, without intermediates. For example, human blood group. There are 4 human blood groups. Types of prints. Types of earlobes. There are 2 types of earlobes, for example, and so forth. Now, number 5. To show the discrete nature of the feature, separated bars in a bar chart are drawn. So we have to draw the bars separately. After we'll see the next slide how it's drawn. And don't join the bars together as for the histogram in continuous variation. So don't join the bars. And there is no normal distribution curve or bell-shaped curve involved. Number 6, discontinuous variation is qualitative in that a characteristic is either present or absent. It cannot be measured, so it's not quantitative, but qualitative. Number 7, discontinuous variation is normally controlled by a single gene, in which usually a single gene consists of two alleles, two different versions. For example, we can use big G to denote the dominant allele and small g to denote the recessive allele. So a single gene means just a pair of alleles will control the characteristic. Number 8, it is not influenced by environmental factors. For example, the presence of dimples. A person is born with dimples or without dimples, and nothing in the environment can affect the absence or presence of dimples in a person. How does the graph for discontinuous variation look like? So the graph for discontinuous variation is very different from that of continuous variation. We don't draw a histogram. We draw separated bars. Separate them and don't make them stick together, right? And this will produce a discrete distribution graph. So there's no histogram and no bell-shaped curve for discontinuous variation. Take, for example, blood group. So we can do a survey in a class to find out the number of students with blood group A, blood group B, blood group AB or O. Some students may have had their blood group tested before, so they know their blood group. Now, it has been found that blood group O is the most common blood group, and blood group AB is the rare one. So plot a graph of the number of students against the blood group. Then here on the horizontal axis, we have to write down the different blood groups, and then plot the graph according to the values shown here. So separate the bars, and also each bar must be of the same width as each other. The bars must be of the same width, okay? And the distance between the bars must be also the same. So we get a discrete distribution graph for discontinuous variation where there are separate bars in the bar chart. Let us now discuss the similarities and differences between continuous variation and discontinuous variation. So this question can come up in the exam. Compare and contrast continuous variation and discontinuous variation, 10 marks. Or you may be given two diagrams, one showing a characteristic which shows continuous variation and another characteristic which shows discontinuous variation. And you may be asked to identify the type of variation and then to compare them. All right? So we must know the examples for continuous variation and also discontinuous variation. Right. The similarities. Both involve differences between individuals of the same species. Right? You must have the word differences and same species. Then both variations contribute to the survival of a species. And another point is that both types of variation are influenced by the genetic factor. Okay, that means there are genes that can affect that characteristic. Now, differences. The definition for continuous variation, it is a type of variation in which the differences in a characteristic are not distinct, tidak jelas. But for discontinuous variation, it is a type of variation in which the differences in a characteristic are distinct, are very clear. Examples: height, weight, body weight, hand span, foot length, or even leaf length. For discontinuous variation, blood type or group, ability to roll the tongue, and fingerprint patterns. Now, how about intermediate characteristics? Continuous variation shows a wide range of phenotypes with intermediate characteristics. That's why there are so many phenotypes. Phenotypes means the observable characteristics such as the height or body weight or the hand span and so forth. Now, for discontinuous variation, it shows a few distinct phenotypes only with no intermediate characteristics. All right. Fourthly, is it quantitative or qualitative in nature? For continuous variation, the characteristics are quantitative, meaning that you can measure them, such as height and weight, body weight. For discontinuous variation, the characteristics are qualitative. They're either there, but the person has it or doesn't have it, and thus usually they cannot be measured. The fifth difference, the type of graph. For continuous variation, the graph shows a normal distribution curve. That means a bell-shaped curve like this. And the graph we draw is a histogram, where all the bars are attached to each other like that. Now for discontinuous variation, it shows a discrete distribution, like separated bars. Must draw bars that are separated from each other. Environmental factors. Continuous variation is influenced by environmental factors, such as for skin, the amount of exposure to sunlight will affect the color of the skin. And for height is affected by the type and quantity of nutrients that a person eats in his food. Okay. Discontinuous variation, it is not influenced by environmental factors. For example, blood type or blood group is not influenced by any environmental factors. Now, genes. For continuous variation, it is controlled by several genes or several pairs of alleles, most probably on several different types of chromosomes. But for discontinuous variation, the characteristic is controlled by a single gene, usually a pair of alleles. For example, one dominant allele and one recessive allele on a pair of homologous chromosomes. Before ending, let me share with you a fun fact which is very interesting. Do you know that there are some people, or very few people, that can form a clover leaf shape or a flower shape or star shape with the tongue, where the tongue is folded a few times, 3 times or 4 times, to form this shape, this unique shape. Okay, so if you can do this, you are one of those rare ones. In my years of teaching, I've come across 2 students who can do this star shape or clover leaf shape. So it's called the clover leaf tongue, and you can find it on the internet. All right, you can search for that term. So this is a rare ability. It seems to be some kind of innate ability. Whether it's genetic or not, I'm not sure, unless somebody does the research on it to find whether genes are involved. So thanks for watching, and do comment, share, like, and subscribe. Till we meet again, goodbye. 47.